You're listening to the FaceTime with Scientists podcast, and I have a great episode for you today. But first, can you guess what the deadliest animal on planet Earth is? I'll give you a second. It's not a tiger, it's not a bear, it's a mosquito. Today I'm talking to mosquito and malaria researcher Cassandra Fieldson, who's at Seattle Children's, and studying the mosquito and its life cycle in order to better understand and treat malaria. Let's go. So today we're here with Cassandra Fieldson at Seattle Children's, right? A researcher studying mosquitoes and malaria. And I'm super excited to chat with you today. I looked a little bit about some of your other research projects and super excited to dive in. But what do you plan to show us today? What are you doing in the lab today?
So today we're going to be going through a salivary gland dissection where we take the salivary gland from mosquitoes, which are already infected with the malaria parasite, and then we collect them so we can use it for our research later. It's a pretty hands-on process, so it's very exciting to see and talk about all the nuances. So to get started, I actually have a bunch of mosquitoes here. I probably have about 50 mosquitoes in my hand that I previously collected before doing this. And how we collect our mosquitoes, our lab is one of the largest insectaries in the Pacific Northwest, which is very cool. And we raise 50,000 mosquitoes a week. And so in doing that, we have a lot of mosquitoes readily available for our malaria research, which saves a lot of people. And so I collected these by using a vacuum tube, and we stuffed the mosquitoes into a bunch of ethanol. And since mosquito respiration was a little bit different than humans or mammal respiration, whenever they come in contact with ethanol, they actually get knocked out and go unconscious. And we can keep the mosquitoes down using ice, and they stay knocked out for this procedure so we don't feel anything. And it's our way that we collect them and make sure that we have all of it. So it's very fun to be holding that many in our hands.
That's amazing. So yeah, I had looked up that you worked in an insectary. Insectary, is that what is an insectary?
Yeah, and insectary is where we raise and cultivate a lot of our insects, which are any sort of arthropods that has 3 different body segments and 6 legs. So things that might be included could be like ants. You can have an ant insectary, you can have a bee insectary, and in this case, we have a mosquito insectary. And in fact, we have a specific species of mosquito insectary because only Anopheles mosquitoes can transmit the malaria parasite. Um, there are over 3,500 different species of mosquitoes. And so we only rate this one that has the ability to transmit it. Otherwise, other mosquitoes wouldn't do the job that we would like to do.
Wow, I think I'd heard of that before, but I didn't realize that like there was that many different mosquito species, and
Yeah.
only you said like around 10%, sorry, feed off blood, and even fewer than that feed off humans. So like, yeah, just this idea that there's plenty of mosquitoes out there. Some even are doing work as pollinators. And I didn't realize that, so that's really cool.
Yeah, a lot of people I talk to, they're always shocked when I tell them, yeah, there's actually more than just one mosquito that's just out there causing all that chaos for the past how many millions of years. And so having the different species, it's really interesting to study, and each of them play a different role in the ecosystem. And so some of them are flood feeding, and then there's some species that don't even need it at all. So it's not all mosquitoes are bad.
So yeah, do you want to tell us, like, kind of show us and tell us what is a salivary gland and why are you studying that? Why are you dissecting that?
Yeah. So the malaria parasite life cycle is super complicated. It's part of why it is so like long standing. It's something that's very difficult to treat because of that. Malaria has been around and evolved with mosquitoes for over millions of years. We have evidence that mosquitoes have had malaria back in the ages of the dinosaurs, which is crazy to me.
Really?
Yeah. So how we found this out was encased in amber, a biting midge, which is a relative of mosquitoes or an ancestor of mosquitoes, had taken a bite of something, and in its stomach contained the malaria parasite. So this is a kind of interesting Jurassic Park moment. So that's really cool.
Wow.
So this had been evolving with the parasite, this mosquito and the parasite have been evolving together for such a long time that they have at least hundreds of tiny little steps in nuanced details. One of them involves going into the stomach of a mosquito after it takes a blood meal, bursting through that stomach of the mosquito, and creating something called an oocyst, which holds thousands of the parasite ready to replicate and go to the next step. And that oocyst bursts and goes and travels to the salivary glands of the mosquito. And eventually, when it's there, that's when it can be transmitted to the next host. And that process takes about 3 weeks. So it's not an immediate infection.
So the
Yeah.
parasite's actually traveling to the salivary gland as like a route of infecting the next animal.
Yeah, it's very complicated and takes a while. So we have to catch the parasite right when it's in that salivary gland. So that way we can use it for our experiments. We can't just automatically have a mosquito that takes a bite of an infected host and then bam, it's infected. We have a little bit of time and process the vector.
Gotcha.
Our goal is to collect the salivary glands, which is located kind of in the throat of the mosquito. If we were to compare it to human body plans, the best we could. They're very, very small, and I will be showing it on a video here as I'll start to dissect mosquitoes. But
Sweet.
they have 2 different salivary glands with 3 separate lobes. And I've been told they kind of look like a clover shape, which is really cool. With the clover shape, they actually had different lobes that could hold different amounts of parasites. I am distributing a few mosquitoes into onto the plate here, and I'm gonna then take my trusty tools. So I have a forceps or tweezers, and then I also have a needle, and I'm gonna be using those to hopefully start working on the dissection process.
Wow. This must take very steady hands.
I cannot drink caffeine in the morning before I start doing this for sure. Otherwise, I'm a little less likely to get what I want from this. I have 1 female mosquito collected on the plate, and on that plate I have its head closest to my right hand or the side with the needle, and then I have on my left hand the abdomen, which is closest to my left hand, which is really cool to see. I'm gonna grab the abdomen of the mosquito's body with those tweezers, and then use my needle to slightly pull the head away from the body. And in doing that, it's a little bit difficult to see, but there are 3 tiny little lobes that I've just removed. So mosquito dissection, yes, take very steady hands. We have to make sure that we're not ripping it apart too much or accidentally squishing it in ways that we don't want it to. So for me, what I do is I usually take my tweezers and grab the thorax, which is the largest body section of the mosquito. That's actually in female mosquitoes where the ovaries are held. And so if you accidentally squish that, you can have all the eggs that are inside the mosquito go everywhere.
Wow.
So I grab that thorax, and then with my my needle, I slowly move the head away from the rest of the body. And it takes a little bit of getting used to it, but you have to learn how to keep the rest of the digestive tract intact while you're separating this mosquito, which is a little bit difficult to do. And then eventually you can sever the rest of the body away from the salivary glands. And I think I should have them collected right there, and I'm pointing at it with the needle. And so for people who are listening along, this looks like 6 little small clover-shaped lobes that are very translucent and small and hard to see. And they have thousands of malaria parasites collected inside of them. So I'm going to put it off the side and keep dissecting, and maybe we'll talk more about mosquito fast.
Yeah, that's amazing. Okay, so do these mosquitoes that you're working with right now, I mean, do they have malaria parasites in there?
Yes, actually, these ones right now that I collected for this have malaria parasites in them. But
Wow.
they don't have, we're not working with a human infectious strain for this experiment. So
Gotcha.
I'm a little less protected by different PPE than I am. Certainly, I'm just wearing gloves, but working with human stage malaria, you have to like wear the whole fluffy marshmallow suit to make sure that you're safe and you don't get you have an extra layer of protection between you and the parasite. But in this case, I feel pretty safe because we're actually working with a strain of mouse malaria.
Gotcha. Okay, so this is interesting because I learned through some of your background that you have also studied birds in
Yes.
different research studies, but that part of that was it your first encounter with malaria that you actually were studying hummingbirds and hummingbirds can have malaria, which I did not know this.
Yeah. So, weird thing about malaria because a lot of human infectious diseases, I feel like I have this make this mistake all the time. I think only humans can get them. I forget
Right.
that there has to be like an evolutionary process to get to the point where humans are infected. And so malaria has a complex cycle. And we know that mosquitoes don't only feed on humans, they can feed on other species. So because of that, we know that malaria has come from different species. And bird malaria is a bit different from human malaria because the red blood cells of birds have a nucleus in them, where human red blood cells don't have a nucleus at all. So this stage of malaria infects not only the parasite, the parasite infects not only the red blood cells, but it infects the nucleus of the red blood cell, and that starts a whole different stage. So bird malaria is even more devastating to their populations than
Really?
human malaria.
And is that something that, like, is bird malaria, I mean, an issue in the United States? Is it an issue mostly in countries where we also know of human malaria?
Currently, I know of a lot of research that's being done in Colombia of hummingbird species that are impacted by malaria. I also know of projects that are happening in Hawaii where a lot of their endemic bird species are being absolutely devastated by malaria. So there's a lot of work towards malaria control with the purpose of conservation, which is a really interesting thing. Yeah.
Interesting. That's crazy.
But
Um,
it was really interesting to see this because I was working with hummingbird respiration, and you could see differences in how hummingbird respirations were impacted if they had malaria versus if they hadn't. Because a lot of malaria's side effects comes from the bursting of blood vessels, the bursting of blood cells themselves, and that just makes it a lot more difficult to breathe. So it takes more energy for you to go through respiration. So very interesting
Interesting.
stuff.
Yeah, okay, so for, in terms of why you're studying malaria and dissecting these mosquitoes and pulling out their salivary glands, what's the kind of like next step? Like, what is your goal in studying the malaria parasite within those glands, and what is the goal of this lab in terms of, is it better understanding the disease? I know you talked a little bit in some content that I've watched of your science communication, just like the better that we understand the mosquito and its life cycle and how the pathogen moves throughout mosquitoes to different, to animals and back again, helps us understand malaria and potentially treat it. But yeah, what is your approach or
What
the
is the next
lab's
step?
approach to, yeah?
So our lab focuses on a few different things. We're pretty widespread with our focuses, but we focus on liver stage malaria. Like I mentioned, malaria goes through a lot of different cycles. So the liver is where it stops first when it infects humans. So it's a very likely vaccine target as a possibility in that direction. We also study something called cerebral malaria, which is what happens when people start to feel the side effects of malaria. It is pretty devastating considering how many people are impacted by it. And then finally, we work on automation processes, like how to create more salivary gland dissections. So you can either work on creating live attenuated vaccines or also creating more research. So we have a lot of different angles, but in this case, for this experiment that I'm going to be doing, I'm hoping to take these sporozoite parasites and infect them into liver cells that I have already plated in vitro, which means not in a living animal or anything. So these are individual cells that I've been infecting with the malaria parasite, and hopefully we're trying to see different different genetics differences in the liver cells if we can find any triggers. So we have two malaria vaccines that exist, but they don't that are already like federally approved and something that is safe to use. But we're obviously working to create more, as there's still 610,000 people who die a year because of malaria. That's a very high number, and we want to reduce that as we can. And so we are trying to have different methods that we can try to create possible vaccination methods. And one of those is through live attenuation, which is the concept of taking the parasite, the pathogen, anything that you have, and destroying it in a way, breaking it a little bit. So that way it doesn't fully make you sick, but when your body is introduced to it, it recognizes it. Because a lot of researchers agree with this concept that you're never going to get better immune response or immune protection unless it's with the pathogen itself. Like your best form of protection is going to be through introducing yourself to the pathogen and addressing it through your body's own mechanisms. So it's
Right.
trying to find different avenues that we could possibly address it.
Yeah, yeah. That's super interesting.
Yeah.
So, I mean, when you're studying these mosquitoes in the lab, and I think in particular, well, back up. You have an Instagram that's and TikTok, I believe, as well, that is very popular as at Biocast, where you show yourself in the lab and you show the dealing with mosquitoes. And I think I saw you do a video where you're kind of holding your hand over the mosquitoes and they're kind of punching their little, I'm gonna call it noses. I don't
They're
know.
called proboscises, but yes, they're noses. They're little mouth farts. They're yeah. It's always funny to hear creative ways to call them, but
Yeah,
proboscis
their
works. Mouth
mouth
part
parts.
works.
Yeah,
Yeah.
pushing up through the fabric and they can't reach you. It was like I love kind of rage baiting the
Oh
mosquitoes of
yeah.
like
Yeah.
is this something in terms of working with mosquitoes in the lab? Do you think of these things as a deadly animal or do you kind of think of them as your pets? Like how do you think? Yeah.
Ooh, that's an interesting question. Like, where do I draw the line with them? I think I've been working with these mosquitoes for about 3 years now. So I see them almost daily. I still see them as a being, but I do have empathy for them and I do care about them quite a bit because each time I work with them, I want them to be successful, either for my experiments, but also you start to work with something close enough, you like recognize its behavior, you start to them really interested and intrigued in how it works and how it functions. But I think that there is that level of they are causing millions or hundreds of thousands of deaths every year.
Yeah.
There's also interesting concepts like how people view insects versus mammals versus other high order creatures. And since insects are very different from humans, it's a little bit easier to dissociate. But my official answer is I care about them.
Yeah.
I think they're really cool. And I, as an entomologist, am really fascinated with them. But yeah, I think that's my final
Yeah.
answer to that.
Yeah. No, I
Yes.
think it's interesting. You also have a bit of a history as like a wilderness instructor. Like you've
Yeah.
like helped people have relationships with nature. How does some of that work and kind of hobby that you do like kind of tied into your research now in
Yeah.
terms of studying these things?
I think I got my start with science very, very young. I'm very lucky about that, where I was outdoors all the time. And because I was outdoors, I was just asking questions and how things work. And for me, that's the stuff that I love. Just sitting there and pondering and asking. I think that's true fundamental science. Like, you just learn more about your world. And my start was through nature. And so it feels very logical for me to sort of guide people those interesting questions through asking about questions in nature. And so uh, I started probably when I was like 15 being a wilderness survival instructor. And for me, what I gravitated towards when I had the chance to teach anyone anything was let's talk about this plant or this really interesting creature and just let people explore with it and get really interested by it. And by talking about these things, it caused me to research them more and want to learn more. And it's what drove me to start my first position in a lab studying birds because I reached out to a researcher who I knew and I asked, hey, you do some really cool work. I would love to understand more about what you do. And through talking to the scientists, I got to go check out their lab, see what their work is like. And I got really fascinated. They happened to have a volunteer opportunity for me. So it sort of changed into me supporting research. So having that pathway where I started with just asking questions to teaching people also how to ask questions and being in nature really led me to where I am now.
Do you enjoy time at the bench? Like, is this, I don't know, is this peaceful or meditative for you, dissecting mosquitoes?
I will say specifically dissecting mosquitoes is very cathartic. If you've never been bitten by a mosquito,
True.
but it also is very peaceful because it's the same task over and over again. So it's very repetitive and you start to see the same things and get pretty good at doing it very efficiently. So for me, it is a bit peaceful, but there are some days where I want to go out and go do field work. So, or be outdoors and connect with nature because I think that's what connects science to a lot of things. And so for me, going out to nature is either my way to reconnect with science and get back to my roots if like I feel like the lab is getting too frustrating, or it's something that I actively go seek out to do. So I'm really excited. In a few weeks, I'll be able to go out and do field work with hummingbirds again, which
Ah,
is I'm very excited about. So they keep inviting me back, and so I will keep going if I can. So I find it's very important for me to find ways to connect with nature to balance out lab work or bench work.
Right. So what
Yeah.
are you now, I mean, you're continuing to dissect, like how many of these do you have to dissect for an experiment usually?
So for this, I'm probably only gonna dissect 25 or so. But on usual experiments, we dissect 200 to even the most. One day we dissected 1,000, which was a long day, but very worth it for our experiments to work out. Um,
Wow, that is a lot of mosquitoes to dissect. Is it,
yeah.
so I'm assuming you can't culture the malaria parasite without a mosquito? Like you can't do it in a dish?
You can't do it in a dish. There's some experiments that people do in vivo where they're able to do it,
Mm.
but our lab chooses to not do that. We're looking to use the sporozoites, and you could ask different questions with what you're able to do. By using the mosquitoes to culture malaria, it allows us to ask questions that are like, how does the parasite first interact with a host? Like, how does it first get to the next stage in its life cycle? Where if you asked questions in vivo, where you might be like working with the blood stage of malaria, you ask like, what are the side effects or the impacts?
Yeah,
So having an insectary like this allows us to ask questions that other researchers can't, because taking the time to raise 50,000 mosquitoes is quite a bit of work. So I feel very privileged to work in a space like this.
that's
Yeah.
amazing. Yeah, I didn't realize that it was kind of, some people raise cells in
Hmm.
terms of like for their experiments, and you're raising mosquitoes for your experiments. But
Yeah.
like you said, being able to track the parasite through the mosquito life cycle, and then you said you're also using that to treat or to infect cells on a plate as well as like whole animals.
Yeah. But our research is specifically working on cells with plates, but there are different avenues for that depending on what questions there are.
What is the next step in terms of, yeah, once you've collected all of the malaria parasites that you need?
Yeah, like what's the next move? So right now we have the salivary glands all flexed. So for those who can see, I have a pile of all of them right here. And what I'm going to do is pick them up and move them into a tube. Most of science is just moving things from one tube to another. And so I'm going to continue with that tradition and pick up quite a few more and move them to our next tube. Is going to be where I crush up all the salivary glands to release the parasite from it. So
Gotcha.
I have in front of me, or in front of us, about 25 salivary glands in a specific cell media solution called Schneider solution. And Schneider solution allows the sporozoites, or the parasite that we're trying to fluff, to stay alive while it's suspended in that solution. So I can't use things like water to put this in, but it does hold all the parasites there and make sure that they continue living. That way we can do the rest of our research. I mean, I'm going to take this is a pestle. I always mix up the mortar and pestle. And I'm going to crush up all the salivary glands inside of the tube. And in doing that, hopefully it releases a lot of the parasites. And um, yeah, and we're gonna move on to the next step, which is to then centrifuge and use centripetal force, which is the downward motion that if you spin something in a circle really fast, it will collect it all at the bottom of the tube. Centripetal force kind of reminds me of the action of, if you've ever been to like a fair or anything and they have those rides where if you sit against the wall and they spin it super fast, you can start to float up against the wall. We're doing the kind of same thing here with this experiment, where we're trying to spin it all down to the bottom.
Yeah, I was
Mhm.
going to ask you. I mean, I'm guessing you have to get a pretty pure sample of the parasite away from the salivary glands or other things that in the mosquito. But like, how important is that like purification process? Does it have to be very pure?
No, that's a great question. That's actually part of what our lab research is, which is how pure do we need to make sure things are, especially if we was thinking about doing live attenuated vaccines, because at the
Right.
end of the day, we're taking gunk and we're using that gunk to do other sciencey things, right? So we want to know how much of it needs to be gunk and how much it needs to be parasite before it becomes either dangerous or it's still effective without having new consequences. So in this case, we want to have majority of it being parasites. For my cells, which I'd be using these with this experiment more, they're very weak. They don't have an immune system to protect them. They don't have anything else like to stop other gunk from interacting with them. So we want it to be very, very fair. We want it to only be the parasite itself. So to do that, I spin them at 1,000 Gs for about 1 minute, and then we grind them up again and again. So yeah.
That makes sense because I guess with the cells, any impacts that you see on the cells, you want to make sure it's just because of the parasite and not because of like other things from the
Yeah.
mosquito salivary gland.
Oh, it's mad at me again. There it is. Yeah, but it involves, well, this next part just involves the exact same process over and over again of grinding our little parasites up and then spinning them down, which is always good. But I can see they're in there, which is exciting because that means I collected the right things and the right things are happening to lead the project forward, which is always good.
That's awesome. Well, this is maybe a good time to ask you, like, what do you wish more people knew about the process of science, as well as, like, in particular, your science?
1 second, the machine's mad at me. Okay, it's less mad at me now,
Okay.
I think. Yeah. Okay. No. They always do this to me. This is also the fun part of science where things don't work like we're supposed to. All
It
right.
reminds me of, like, in Hail Mary where the centrifuge, he's like,
Oh, yeah.
everyone was, like, mad that he put 2 tubes on one side of the centrifuge,
Yeah.
but
I saw that. I was like, oh, I think it would just explode anyway. But luckily, I don't think this one will explode. Otherwise, I wouldn't sit that close to it. So I trust it and it's well balanced. Something that I run into a lot, or the biggest misconceptions that I've heard either through comments or anything like that, is that scientists are trying to do like the most craziest option or the most insane thing. A lot of times scientists are looking for the same goals that you'd want. They're not the bad guys. Um like so a lot of our processes are very repetitive and end up trying to figure out the exact same thing, or trying to be as precise as they can with either how they do things or how they speak about their work, which can kind of have some different uh yeah, but there's a different communications barrier, I think, between scientists and everyone else because you're trying to be the most precise. I think a lot of scientific language is not very definitive. It avoids using words like I'm 100% sure that this is going to happen. It can often make us seem like we're very unsure about what we're saying, but no, we're just trying to be exact because there's always that chance that something else might happen.
yeah, I think, I've talked to some other scientists who say similar things, like, yeah, just the way that we, in doing science, we're trained to talk in a particular way, which is being very precise about our language and what we mean when we say, particular elements of jargon. It isn't just a hard word for the sake of being a hard word. It's like a very precise meaning to that word. But when we're communicating externally, people want to know, okay, like what does that mean in terms of impact, or what does it mean for me, or what does it mean, in terms of action, but we don't always know enough to get to that step. So I think like communicating that next step of like, what should we do, or what the impact is, but also being true to the science is sometimes difficult, and maybe people,
Yeah.
not everyone realizes like how difficult making that interpretation and that step is, and why scientists are nervous to say that next part, because sometimes it's like the answer is we don't know.
Mhm. Yeah. No, I think that's really well said. And that's why I think the work that you're doing, talking to scientists and having these conversations is so important. So really hats off to you. This is the important stuff to get those things out there. So I appreciate it a lot. I was really excited about this. So
Awesome.
yeah.
Yeah. Yeah, me too.
Yeah.
Well, so you've talked about in your science communication, or yeah, because you do a lot of science communication on social media yourself, you really want to kind of spark people's curiosity or make science more approachable. And you mentioned that a little bit, but how do you think about that in terms of, yeah, making it more approachable?
I think the ways that I want to make it more approachable is I want to make it a little less I think there's a lot of bad habits in scientists when it comes to communicating science. And I fall through these traps all the time. And I even catch myself talking about them today. I'm like, oh, I could have said this better. But always trying to talk about science from the perspective of myself when I first learned about these things. Because
Hmm.
I think it's not really fair to start talking about science and expect everyone to be on exactly the same page, because it has taken me years to figure out this stuff. So I shouldn't be expecting people to just know automatically what a sporozoite is, what a hepatocyte is, or that malaria, another name for it is plasmodium. All these things are important and true, but if I'd asked myself that like 3 years ago, I would have given myself a blank stare. Or so I've been trying to make sure that it's just communicated to me as if I just learned about it. Is the thing I keep in my mind. And other things is I'm a very visual person. So I pride myself in trying to get the most like visually easily to understand things that I see. So if I'm starting to understand, oh, the sporozoites burst from an oocyst, which I talked about earlier, I try my best to try to get footage of an oocyst bursting, so you can really see that rather than just hear someone say those words.
Yeah, that's
Yeah.
amazing. That's super cool.
Um, all right, so now we're going to talk a little bit about the scientific process that I'm working through right now. So I've ground up the sporozoites a bunch of times. I've spun them down a bunch of times. And right now they are all collected at the bottom of this Eppendorf tube. And what I'm going to do is collect something called the supernatant, or also known as like the liquid that is above all the other materials. So super like above and natant, I'm not necessarily sure, but I'm pretty sure it has something to do with liquid. So I'm collecting all that liquid above because that's where the super light sporozoites are. So all that malaria parasite is located right in that liquid, and I want to collect it. So hopefully, that's going to be what I can use for my experiments. So I'm going to use my nice pipette to move one from the other. So, mm,
So one thing that reminds me of is that I've worked with scientists before in the lab that mention, and it kind of still goes to earlier you talked about like automating some of these things, that there is a very human element to science, even like the pipetting technique of like one scientist being different from the other scientist. And when it comes to science and we're trying to like isolate different factors and make sure everything is standardized so we can compare like experiments in your lab to experiments in another
right.
lab. Um, yeah, is that something and you mentioned the automation, how what is
Um,
your work on that related to?
yeah, so our work related to automation is we have been trying to do a few new techniques. So first, we are working something called a mosquito salivary gland dissector, a robot that would dissect our mosquitoes for us, which has been something we've been working on for about a year now. We have a robot built and patented, and I'm very excited about it. But
Really?
we're now working on like validating it, making sure it can consistently get those results, that we're not creating a product or a thing that a lot of researchers could use and be like using, and it wouldn't be as consistent for them. So having that consistency allows for our experiments. We're comparing things that have actually changed inside the experiment instead of human error, which is really important. And obviously, we're all humans. We're all going to make mistakes. But I think it's just more trying to make sure that we can kind of catch the inconsistency so you know what you're comparing.
Yeah.
So, yeah, very
That's
fun.
really interesting. I mean, a robot to dissect mosquitoes seems like a pretty good use of a robot, if you ask me.
Oh yeah. When I talk about this robot, I don't know if you saw this video. It's a few years ago, but they did surgery on a grape video. It's nothing like that. It's a lot of different tubes that route the mosquito in a certain way. But
Gotcha.
whenever I say mosquito dissector robot, I think that's what first pops into people's mind is a very technical robot that does exactly what each hand movement is. But it's a little less precise than that, and we're trying to make it as fast as possible. So very fun.
Gotcha. That's super interesting.
Yeah. So for the next step in my experiment, what I'm doing is I'm moving the material into something called a hemocytometer. And that is a fancy way to describe uh, a way for us to count how many parasites there are and make an estimate. It also allows us to view the parasites, and it has a grid so that way we can count each individual square and see how many of the parasite are located in that square. And then based on that small, tiny amount, we can make an estimation that the rest of the liquid that we have is going to have X number of parasites in it. So it allows us to know about how many parasites we are looking for. So I'm moving to that to now, and then hopefully I have another setup all set up, and we can take a look at those parasites on a microscope. So
Oh my
I'm very excited by it.
goodness. Yeah, that's
So
amazing.
yeah, I again for me, like I said earlier, I really connect with any sort of visual thing where I can see, wow, it's real, it's really there. There's truly a parasite there. That is one of the things that drew me into science in the first place, and is what makes science so interesting to me. So I try to have as many of those opportunities as I can.
Is there something that I don't know has surprised you that you've learned that you didn't anticipate doing this research, or like what are some myths that about malaria or mosquitoes that you have yourself myth busted over the
Oh
course of your
yes.
learning?
This is one of my favorite ones to talk about. I know you'd mentioned earlier the one specific video that like a lot of people have seen where there's a bunch of mosquitoes going for my hands. And that is a very notorious video. But all the mosquitoes are attracted because mosquitoes are really good at finding their hosts. They have three different senses that can do that. They use something called chemosensation, which is their ability to smell, so they can smell the specific odors. They have the ability to recognize heat, or through thermosensation, they can recognize if your body temperature is there. And then finally, they can detect CO2. Those are their three main ways to detect things. But a comment and things I consistently see is, oh, they know that you must be an O negative blood. You must be an O type blood.
uh,
And that must be why they like you so much. But your blood is not aerosolized, or at least I hope it isn't. If it is, please go talk to the doctor. I'd hope that your blood isn't out there. So, and that's a big misconception that I often see and hear with science in the mosquito world is that your blood type impacts things. And now there are some studies that show some people have a slight statistical significance in their ability to attract mosquitoes. It's not their blood that's doing it; it's their odor. So, like how they smell. And so, yeah,
Interesting.
that's my little myth busting that I like to do if I can.
Yeah, I've
It's
definitely
always
heard
weird.
that. Well, in our family, we just say like you have sweet blood or not.
Oh yeah, yeah. But that's like the one big thing that I end up hearing those. All right, and now I have, and currently I will send you the footage for this, but at the current moment I have a parasite on our screen, so that means we successfully collect a parasite in doing this.
Woohoo!
What we're looking at here is something called a sporozoite, which is the stage of the parasite that is excreted from the mosquito salivary glands, and it goes into the host, and its only goal is to make it into the liver. And it does that through going through the blood vessels until it's eventually taken to the liver. And the liver is really good at screening for like, any sort of toxins or anything, so your blood likes to filter things through it. And that helps the parasite because it eventually infects a liver cell and uses that cell to replicate and then infect the rest of your blood. So yeah, this is one of the first things that I saw when I was studying malaria. I was like, wow, this is real. This is something that's in front of me. And I I try to show it to people as much as they'll listen. So yeah.
That's amazing.
Mm.
That's interesting that the parasite kind of goes to where you would think it would go if your body's filtering out bad things into the liver, but then to then infect the liver seems like a very smart thing for that parasite to do.
Yeah. No, but it's very necessary for their system. If they don't make it to the liver, they don't end up replicating, and uh, the host doesn't end up feeling any of the symptoms until after the parasite bursts from the liver and makes it into the blood stage. So when it's in the liver stage, because it only infects like maybe at most 10 liver cells at a time, or 10 different liver cells per infection at most,
Oh
it
wow.
ends up not being something that you feel or you're feeling symptoms for. The symptoms come when all your blood cells burst after they're infected with the malaria parasites. And that's when you start to feel anemia and all the gunk that's birthed from those blood cells makes it to the rest of your body. Your immune system starts to react. You start to get feverish and sweaty. And that's when a lot of complications arise for a lot of people. So,
Gotcha. And then I guess from there it's making it to the blood, and then that's where
mhm.
another mosquito that would bite you could transmit. Is that something that is common in the way that malaria is transmitted? Is the mosquito getting it from other humans that are infected, or is it usually from animal?
So that's some really great questions. The most deadly and most impactful malaria parasite that we have are malaria vivax and or Plasmodium vivax and Plasmodium falciparum. And falciparum is the most deadly, it causes the most complication, but those are transmitted between humans. There are some parasites out there, like the Plasmodium noelzi, that is can be transmitted from primate to human, or human to primate, or human mosquito primate, or primate mosquito human,
Mm.
which makes it very interesting because it doesn't, there's still that jump happening. And that's like one of those ways that you can think of the evolutionary history of how malaria has evolved and how it's not only centered around targeting humans.
Interesting. Oh, so when you say the most deadly malaria is transferred human to human, you mean like
Mhm.
direct human to human, not
Oh,
human to mosquito to human, or
I should have been a little more clear about that. From human to mosquito to
gotcha,
human.
gotcha.
The only way that I could see someone getting malaria transmitted to them from another human is either if you're having a blood transfusion, or yeah, that's the only other way I can think of it.
Right. And then I know that there's concern with the changing climate and warming places, becoming, well, yeah, different parts of the world changing in terms of their climate and mosquitoes being able to move. Like, is there concern for malaria parasite, like it's spreading in mosquito populations to a larger part of the world? I know we worry about in the US mosquitoes coming from maybe more south and warmer climates.
That's a great question. And a really big thing on malaria researchers' minds, because of the fact that malaria is transmitted by mosquitoes, it's controlled by how far the vectors can get. And with climate change, we have more habitable areas for mosquitoes moving further and further up north. But we also have to keep in mind that it can only be transmitted by Anopheles mosquitoes. So it's dependent on where those mosquitoes can exist. And also, we have this really cool thing, which is like we have transportation of planes and other things that can cause a lot more travel of the parasite, which is a concern. Mostly the mosquito can get to farther places. I mean,
Right.
one of the examples I can think of uh, how mosquitoes are continuing to spread. Um, Before October 2025, there used to be two places in the world that were mosquito free. That was, I believe, Greenland and Antarctica. Well, as of October 2025, the first mosquito was found in Greenland. So we now see this
Interesting.
spread further and further north, because mosquitoes have more abilities to live in other places, because humans can transport those mosquito places, and also because of the fact the temperatures are increasing or changing because of climate change.
Right. I think I had heard about that like the mosquitoes were making it to the poles, which is very concerning.
A little scary
Certainly,
here.
I'm in Louisiana, so we have plenty of mosquitoes
Yeah.
here. Do
Oh,
you
yeah.
have like advice for people in terms of just like, I don't know, how to avoid
Oh,
mosquitoes?
yeah. No, that's a great question. First up, cover up. If you have like less areas for mosquitoes to get you, you're less likely to be bit. It's just like if they can't get you. But obviously, no one wants to do that all the time. So, like I talked about earlier, what attracts mosquitoes to their host is your body temperature, your how you smell, and also how much CO2 you let out. We obviously can't stop letting out CO2 because that would involve stopping breathing, so we're not going to change that one. Your body temperature is pretty difficult to change, and I wouldn't recommend changing that. But what you can change is how you smell and the odors that you have. So a lot of times different sprays are recommended for that. So I believe a lot of people recommend at least 30% food if you're in really heavy mosquito areas. But we also want to, there is a cost and reward of your own safety. So mosquitoes are very difficult to look around. Mosquito nets are always a good thing to have. And making sure that you just have as much protection as you can from mosquitoes is my best recommendation. But of course, they're very pesky and they will get all over the place. So try your best to swat them if you can.
Yeah, awesome.
Yeah.
Well, yeah. Are you finished with your experiment, your process for today?
For today, I am finished with my experiment, so I have nothing more to perform on camera. But yeah, there will be more steps, but that's for weeks on from now. So there.
Yeah, that's amazing. Awesome. Was there anything, I don't know, that we didn't chat about that you would want listeners to know or watchers to know?
I think that if there's anything I want listeners to know is that some of the most interesting things in science could be some of the most repulsive things to a lot of people. I study mosquitoes, and most people, their first reaction is, ew, that's disgusting. But a lot of times they're the weirdest creatures out there, and they have the most scientifically interesting things. So I think continuing to ask those really interesting questions and just continue to ask why. Yeah, this is my big thing to tell people. But yeah. Also, I post a lot about different size communication things. You can follow me at bio.cast.ification. There's my play on words for bioclassification with my own name involved. And yeah, I'll be posting more there. So if you want to follow me along that way, yeah.
Awesome. Thank you so much. It was a pleasure
Yeah.
talking with you today.
Yeah. Thank you. This has been so much fun. I love talking about science with people. So this is a treat for me.